I will not have a very long speech, and from the beginning it was my intention to talk about proteochemometrics, which Andreas mentioned, which is a model and technology that we first published in 2001. But I thought that I rather should start my talk with the origin of the term pharmaceutical bioinformatics and why we actually came to organize this conference. So bioinformatics is of course a field of understanding biological data, which we do mainly by computers and computations. And bioinformatics is the origin of bioinformatics is fairly young, and it came mainly with sequence data and the need for analyzing sequence data. And this has, of course, evolved into many different fields, as we know, such as genome, proteome analysis, structural bioinformatics, metabolomics, etc. In pharmacy and chemistry, of course, Chemioinformatics is another very important area, and it has a very much earlier origin. And it actually started with the indexing of chemical data with the chemical abstracts, which was just in written format, and then in later on when computers came in database format, which became the CAS index. And from that on, indexing of chemical data and organizing chemical data involved in the field of Cheminformatics, which incorporate or include many fields such as QSR, computational chemistry, molecular modeling, dynamic simulation, Dawkins, etc. And these are, of course, techniques that are in wide use among the pharmaceutical companies and pharmaceutical developments. The proteochemometrics actually came because I'm basically from the beginning a molecular pharmacologist and experimentalist. And it came just by a serendipity finding, and we were interested in doing, we had created chimeric receptor proteins. We had cloned these melanocortin receptors, and to gain insight into the binding modes of how peptides actually bound to these receptors, we had created chimeras of them. So they are constructed from, in this case, two different types of receptors, the melanocortin 1 receptor stretches and the melanocortin 3 receptor stretches. So they are artificial proteins. And what we had done was also to construct small libraries of peptides that were actually the alpha MSH peptide, which is a measurable one, and the MSO5, which is the artificial variant that we had selected by phage display technology as a very selective one for the MC1 receptor. So the question is, how did this selectivity come through? So what we did was to create chimeric peptides. So there are chimeric receptors and chimeric peptides from these two variants. So there are only then 4 chimeras peptides, and there are, I guess, 30 or so chimeric receptors, and all of them were tested. Every species of this peptide were tested for every receptor, which created a set of data, which we then encoded using a binary encoding, a free Wilson type of encoding. So there are number 1 for one type of receptor stretch, and a number 1 for another for the peptide stretch, and a -1 for the other one of the respective species. And when we put this into the computer and did the PLS model, this came out as the first data that we saw. And for me, being a biologist, coming from the biology, this made me kind of crazy, because I bounced up and down for 10 minutes, I can say, because they were on the line. So what we can do is predict the observed versus the calculated from the model, but they also cross-validate. I don't go into all these cross-validation details, but they are statistically valid models. Sorry. And this meant, of course, that we could actually merge biology and chemistry by encoding things in a chemical sense from peptides, and the biological data in a chemical sense by just numbers. then we could actually model them in one unit. So from that on, we started to investigate this way of studying interaction more widely, and we did a lot of studies, which I will not go into all of the details here, but the aim was to validate that yes, this is a possible way of getting models which are useful in some sense. So we had this, where we had 4 different types of deep protein coupled melanocortin receptors, and we made chimeras among them to create a wider library than more chemical variation here. And one of the studies aimed to see if we could model biology, the biological portion, and do a priori prediction of the outcome of an experiment. So, and this contained a small library of peptides and the library of the receptors, and we made a model. And I don't go into the technical details of this model, but the model was then used to predict which amino acids are actually causing an increase in affinity for the melanocortin 1 receptor for alpha MSH. And actually it pointed out 7 hotspots, amino acids, which were the important ones. I condensed this a little, I don't go into a lot of details here, but essentially 7 amino acids indicated by the model that these are the ones that if you mutate them in a certain direction, they should increase affinity. And the red ones are the predicted increases. And then we actually afterwards made the point mutations, and actually in all cases the direction was as predicted. So this convinced us that actually there was a possibility to model the biology. In the same way, we made, these are the results of the model. Actually, the predicted calculated is observed, and the cross validations and things, so on. I don't go into the details here. In the same way, in actually the same manner, we said that okay, we shall predict compounds that show a certain affinity, preferably higher affinity for a receptor. And then we had a library of compounds, and then we predicted new compounds and synthesized them, and actually we were able to show that we can improve the affinity of a compound by a priori predicting from the model. And these are then based on chemical descriptors of amino acid chemical properties and descriptors of chemical properties of compounds. So this convinced us that actually this way of merging biology and chemistry in one unity worked. But when we started to communicating our results, then we encountered major problems, especially among the biologists. They regarded this as non-believable and very difficult they didn't comprehend it. So what we said then was that actually what we lacked was education. So we started this field of pharmaceutical bioinformatics, we call it that, and started education in the field. And this has resulted then we wrote a textbook also in the field for the purpose of the course. And now we educate about 500 students in pharmaceutical bioinformatics, as we call it, every year, mainly through internet-based courses. And this led forward to the definition that I made for pharmaceutical bioinformatics already in my introductory speech, where actually the sciences, informatics, algorithms, and computations apply to the field of drugs in relation to their interaction with the living organism. Because I'm a pharmacologist, basically what we are studying in pharmacology is the interaction of chemicals with the living organism, and this is the core then of actually pharmacology and the use of pharmaceutics. So of course we can use this in improving aiding drug discovery and development, understanding drug actions in the living organism, improving pharmacotherapy, improving safety, etc. And it's our hope that under this umbrella of pharmaceutical bioinformatics we can gather future conferences where we can advance the field and have a platform for mutual exchange and development in this field. I'll come back to this later. And before I left, I found this one because I thought it was quite funny, so I could paraphrase it. We are surrounded by scientific problems, which is excellent because we can do research in any direction. Thank you.